This presentation on MAPpoly is sponsored by the Tools for Genomics Assisted Breeding and Polyploids Training Workshop and funded by NIFA, the National Institute of Food and Agriculture within the USDA Specialty Crop Research Initiative, and was held on January 14, 2021.
Welcome to the MAPpoly training section. We had a very productive section with Peter, and now I will show you some features of our program or our package. And so this is just an outline of the things we are going to see. And just right off the bat, I would like to acknowledge some people here. First, I'd like to thank Gabriel Gestera, which is helping me a lot programming MAPpoly, and Gui as well. My former supervisor in Brazil, Augusto Garcia, Craig Yancho, our collaborator at North Carolina State, and my supervisor nowadays, Zhao Bang, which we have very fruitful discussions about all the stuff that we implemented here. And just as an initial note about MAPpoly, so when we start to implement MAPpoly, we aim for haploid levels, because the first species that we used to work was sugarcane, which is a high heterozygous and haploid level species. And after that, I went to North Carolina State, and then we start to work with sweet potato, which is an hexaploid. So we always aim for haploid levels. But of course, if you manage to figure out how to do for hexaploids, so you manage to do for tetraploids as well. Just let me start my stopwatch here. Set. Yeah. So, and everything that I'm presenting here today is published in this paper here, the theory. And the principal goal of MAPpoly is to build the map, but not just build the map, but also obtain the conditional probabilities for the QTL analysis, which is the next phase, the next part of the process. And here we will not model double reduction, because as I said, we aim for haploid levels, and then it's very complicated to model double reduction in haploid levels. But we will use MAPpoly without this feature. And yeah, so first, we have two versions of MAPpoly. One is the stable version, which is available on CRAN. You can just install MAPpoly using the install packages within R. And we also have a development version, which is every week I have an updated and a new feature in MAPpoly. If you want to install that version, you just use DevTools, and you use this install GitHub function and point to my GitHub page in MAPpoly. Okay, so this is just the installation. It's pretty straightforward, and this is the mapping process in MAPpoly. It's a little bit different from Polymap R, as we will see. But we also have the filtering process, we have the 2-point analysis, the grouping. After that, we order the markers. The group is to splitting linkage groups. After that, we order the markers. We perform the phasing. We take into account the errors using the hidden Markov model. And then after that, we computed the genotype probabilities with those, with the phasing map, or with the map generated with the modeling error. And then we can compute the homolog probabilities, or the haplotype probabilities, and you can compute preferential pairing profiles. And using this information, you can perform your QTL analysis. And also you can have your map summary for publications and so on. So we have some types of data that we can import in MAPpoly. So there are two types of files that have a very specific format, and everything is explained in a tutorial that I will show you in a minute. But you also have CSV files with a specific format. You can import data directly from MAPpoly with dosage probabilities. You can import VCF files with discrete dosages and also with dosage probability, taking into account one of the things that you have in VCF files, which is the PL feature. So MAPpoly can upload data from UpDog, PolyRed, and also it can import data from MAPpoly R, datasets and maps. So if you can keep this conversation between the two softwares. And basically for the dataset, you have your dataset, and this is more or less object-oriented. So if you have your dataset and you load your dataset, or if you read your dataset from an external file, it will generate MAPpoly data, okay? So I will change to my RStudio, and I will try to show this. And before we start, I just want to point out the GitHub page for MAPpoly. So if you want discuss something, or if you want a new feature that you might want to use, you can open an issue here in this place here. And as you can see, there are 6 issues that were closed, but you can just ask, oh, Marcelo, we need something in Polymap, or there's a bug in a function, just right here, and I will try to answer as soon as possible. But here is the main page with the code. Everything's open, and the important part is the vignettes part. So everything that I'm going to show you today and more, it's in this link here. Okay, and this is the tutorial for what we're going to do today. As you can see, there's not just we use an output of MAPpoly today, but as you can see, there's an explanation for all other formats here, and we have much more than I'm going to present. Okay, so let's start here loading map. First, yeah, I was forgetting. So the folder with everything that we need to do this analysis today is available on the polyploids.org website. And we are going to use this. There are 2 folders here, one with Exaploid data. This is Exaploid sweet potato data. And here we have our tetraploid data. This is potato data. And if you download this folder from our website, you can follow the scripts here. And yeah, so first, let's load MAPpoly here. And as you can see, so this is the current version, which is 0.2.1. And if you want to download the version from GitHub, today we have the 2.2. And there's some new features, but it's pretty much the same program. Yeah, so the first thing I want to do is to set my working directory to the tetraploid folder. And there you go. And then our data set is actually on the internet, and I'm going to use a nice feature from R, where you can download the file from the internet. Okay, so this is in my GitHub page, and I have this in this address. So you can also click on this and copy and paste in your browser and download this, but let's do it within R. So we have the address, we have a temporary file here, Just let me show you. It creates a temporary file in a specific place in your computer. And then you're going to download this data set, and I'm going to discuss. Yeah, so we have here our data set. It's a zipped file, and you can unzip using the unzip function. And yeah, so, and there you go. We have here our B2721 scores.dat. For this particular data set, I have the script that I used for the snip calling here, right? Which is using MAPpoly, and we are going to load this data set using this function read MAPpoly. Okay, just let me load this. So there are some arguments here. The first one is actually the input file. So this is a potato biparental population. We have two parents. One is Atlantic, and the other one is B11829. And in this case, you need to point to the input file. You have deployed level here, which is 4. You need to tell to the program the name of the parents. Here is the probability threshold, meaning that if the marker is, if the specific data point has a probability lower than 0.95, it will be considered as missing data, okay? Now you can change this, but we learned that for tetraploid, this is a good number. And for specifically this data set, this is a good number. So maybe a little bit stringent for other species. For instance, in our hexaploid map, we use 0.75 here. And here is an example how to load VCF files as well, if you are interested on that. But the important thing here is that if you use any of the functions, of the read functions, it will generate the same object. It will generate an object called MAPpoly data, which will contain the same information, okay? So in this case, we have, in this data set, we have deployed level, we have 156 individuals, more than 7,000 markers, number of informative markers. So this is removing the monomorphic markers. So more than 90% of the markers are polymorphic. And at this point here, the function filtered the non-conforming markers, meaning that there are markers that are impossible to happen in the offspring. And also it performs a chi-square test for all the markers in the population. So, and we can check that. So as I said, the class of this object is MAPpoly data. And if you print that with the detailed equals true, what we have here is the same information we observed before. So the number of missing markers under this probability that we this is similar from what Peter showed us. Here, sometimes you have duplicated markers or the markers that carry the same information and they are not useful for the linkage analysis. And here we have a table with all possible combinations of dosage in both parents, and also the number of individuals in each one of these. So here we have a summary of our data sets. So we have the number of markers for each one of the classes in our offspring. We have the markers here and individuals with a color code. Each one of these codes is a specific dosage in the offspring. And here we have our chi-square test for the polysomic segregation. Yeah, so one of the things that we can inform MAPpoly at the beginning is the position of each one of the markers in reference to a genome, right? So here we have the potato genome, and in this script here, I explain how to merge the data set from the positions of for each one of these SNPs from the public potato genome with the MAPpoly data. But if you are using, for instance, a VCF file, and if this information is contained in the VCF file, it will automatically load in MAPpoly and also with other formats. But in this particular case, since you are using FitPoly, I'm just running a script to concatenate the information from the potato genome in our data. So when you print your data now, you see that you have the number of markers per sequence. And this sequence here means each one of the chromosomes in potato. And we have 274 SNPs that were not assigned to any chromosomes. And the sequence here, this is the chromosome. Sometimes you have I put sequence here, but in this case, since we have the complete genome, we can use the chromosomes, right? So we have our data set loaded here. And again, if you want to see the other types of data sets that you can use, you just go through this tutorial here. And as you can see, we have examples for all possible data sets, including PolyRed, FitPoly, UpDog. Yeah, so it's this one. Okay, so the first thing you might want to do is to inspect a specific SNP, right? So let us take a look in SNP 738. So this is the position of the SNP in the input data set. So, and here, as you can see, there's some information about that specific SNP. Since we have the probability for each one of the SNPs, we have the distribution for each one of the individuals here, right? So in this case, we have a duplex in parent 1, duplex in parent 2, with no missing data. This is the p-value for the chi-square, the chromosome, and the position in the chromosome, and the probability threshold that we used to load this SNP, right? So in this case, this is a very good SNP, as you can see. For instance, in this first row here, we have a SNP, one individual in the offspring with probability, with dosage, which is a nulliplex, and with a very high probability, and so on for the other individuals. And you can print this marker to see the frequency in each one of the classes. And this is the expected polysome segregation. In this case, I'm just showing you another SNP, which is also duplex and duplex, but this is in chromosome number 4, and this is the position in chromosome number 4. But as you can see, the probabilities for some of these SNPs are not that high, and it seems to be called we lose the data set using 0.9. As you can see here, some of these SNPs are considered missing data, right? So if you use a smaller probability threshold when loading the data set, some of these SNPs would be also considered not missing. But in this case, as I said, we are going to use a very stringent threshold. And if you're curious to know what is the frequency or the expected frequency of a marker in your population, for instance, here we have a tetraploid with a simplex in one parent, duplex in the other parent. You can use this function, segregpoly, and as you see here, this is the expected frequencies for SNPs. And you can do this for very high ploid levels. I know this is weird, but you can do something like this for the dodecaploid with the first one with those 10 and the second with those 2. And then you have this weird segregation here, okay? So this is for the loading part. Now let's do some filtering. And also in MAPpoly, similarly as Peter showed us in PolyMap R, the filtering is a very critical part of the linkage mapping. So here we are going to use the first function is the filter missing. So we are going to base our filter in missing data, okay? So in this case, I just use my data and the type of filtering. So here I'm filtering based on the marker. So if the marker has more than 5% in this case of missing data, I'm going to remove. But we can do this interactively. And in this case, if I use 0.5, I would remove 1,230 individuals. I can change this to 0.1, for instance. So it will be less stringent. I will remove 800 markers. But let's go back to 5% of missing data, okay? And when you're done, you just type yes. And then we want to do, now we're going to do the same thing, but based on individuals. Sometimes you have an individual with a lot of missing data. And in this case, I'm just going to show you here, we have 156 individuals. And here I'm being very, very stringent here. And potentially we could use all these individuals here. But sometimes in our exploit data, which will be available and you can try yourself, you see that there's some individuals that had some problems probably with the amplification of the DNA, so you can't see any of the markers for that specific individual. But here, in the same way we did, you could use a different threshold, but let's stick with the threshold of 2.5% of missing data. And just type yes. And in this case, I will plot my data set again. And as you can see, if you compare this figure to the previous figure, you see you have less black dots here because we removed a lot of missing data. Let's see how many markers we have. We have 5,300 markers in this filtered data set. Another thing that we can do is to use filtering, we use segregation to filter. Because if you have an auto polyploid species, you have an expected segregation, and we can use that with a chi-square test to remove some markers. And that's what we're going to do here. So I'm just using the threshold, which is an approximated Bonferroni correction. I'm going to use 0.5, 5%, and I will divide this 5% by the number of markers for the threshold. And as you can see here, remove these markers. If I choose this threshold, I would exclude 350 markers. Again, I can change this number, but for the purpose of this tutorial, I will keep this number. And again, if you, the algorithms here, as you see, they are very robust. So if you want to be more or less stringent in this filtering process, it's okay. But the thing is, if you have less information or if you let the program deal with less informative markers, it will take more time because it will rely on more on the hidden Markov side of the algorithm, which is more intense. But and so here we are being very stringent. And as you can see here, I have S is an object of class MAPpoly keytest.sec. And so here now we have our data filtered by missing data and segregation. Yeah, so the first step, the next step here is to make a sequence of markers. This is a very important object in MAPpoly, and I'm going to show you here. So this is just the graphics that I just showed you. And for those of you that use OneMap, which is another software that we developed a long time ago, this function was inspired on that software that was inspired on MapMaker, which is a old school mapping software. And basically what we do here is to use an object, and then based on an argument, you make an ordered sequence, okay? And for instance, if you have your data, you can use this function to make a sequence of some markers in this data set. If you have a group that I'm going to show you in a minute, but if you have a group in MAPpoly, you can make sequences of that group and so on. So this is a very flexible function. And here I will use this function to make this sequence. So here, as you can see, when I make this sequence, I'm assuming that there's some sort of order in this sequence, okay? It doesn't mean that the markers are ordered according to the genome or according to MDS, but they are in a certain order. And when you build this sequence, you have the MAPpoly.sequence, and you can also plot this MAPpoly.sequence, and you have a similar graphic. And here, as we observed in the when we load the data set, so here again we have the number of individuals per dosage combination in the parents, again our offspring, and these are the chi-square tests for all the markers. And just showing you the functionality, the make-seq MAPpoly functionality. So here, for instance, I want to make a sequence of with the first 30 markers in my data set. I can use this. And then you have 30 markers. They are distributed in these groups here. And this is also, since we have the information of the genome, the potato genome, we can use this argument as we can use seq and the name of the sequence that we want. So here, since sequence means chromosome, here I'm going just to gather the for the chromosome number 10 in potato using this function. And as you can see here, I will just have chromosome number 10, 285 individuals. Beg your pardon, is 285 markers. And we can also plot this, as you see. So these are all markers for chromosome number 10, okay? Yeah, so since we have a sequence of markers, what we can do with this sequence of markers is to compute a recombination fraction between all the markers within this sequence. And what we usually do is to load all markers that are available in our data set, and then we compute the pairwise recombination fraction between all markers, right? And in this case, I had, with this code here, it will do that. In this particular machine, which is a Linux with, as you can see here, we have 16 cores. It will take about 14 minutes, right? But as I said, if you have a sequence, you can compute for that sequence. So here I'm going to choose just to compute this recombination fraction within the sequence of chromosome number 10. In the same way that Peter showed us, we are going to select some virtual cores here, virtual CPUs. In this case, I have 15 virtual CPUs. I'm letting one just to use for other stuff. And here is the function to compute the recombination fraction between all markers in that sequence. Here I put the number of CPUs, and here we can use two types of computations. You can use the discrete dosage calling, or you can use the probabilistic dosage calling to perform this recombination frequency computation. And in this case, we are going to use the discrete one, and it will take about 10 seconds, I believe. And there you go, 7 seconds. And here, as you can see, we computed all the recombination fractions between the 285 markers within this group, okay? And just to show you the concept of linkage in polyploids. So here, I have a list in this object with all pairwise combinations for each one of the markers of the pairs of markers. Just to show you here, this is one output for that type of recombination frequency estimation. So in this case, we have, in this example here, I believe is the same example we have here or not. It's not exactly the same, but still, in this case, we have a triple or a triplex marker for one individual, triplex marker for the locus, for duplex marker for the locus in parent 1, duplex marker for first locus parent 2, duplex markers and second locus in parent 2. So there are several possible combinations in which these variations here can be disposed or can be arranged in these homologs. And those are all possible combinations, right? What I'm using here to describe these combinations are numbers, which means that how many homologs these variants share. So in this case, we have 3 and 2, right? So this is 3 and 2 in this table here. We have several possible linkage phase combinations here. And in this object, in this 2-point object here, we have the combination for all possible markers. And since this is a list, I can show a part of this list using this syntax here. And then what I also can do is to plot the information for a specific combination of SNPs. In this case, between SNPs 1 and 2074. And as you can see here, I have the LOD for each one of the linkage phases for the recombination, for the linkage phases and the associated recombination fractions for that specific linkage phase. And this LOD here in the lower part is the LOD for linkage. As I said to you, I did this just for the chromosome number 10. But in reality, what we want to do is to compute this recombination frequency between all markers in your data set. And you can do this using this syntax here. So instead of using as in chromosome, as in the whole data set, as we defined it before. And the results for that is actually a list. But in terms of recombination fractions, we usually display that in a matrix format, as Peter showed, and as also I showed in my linkage talk. And for doing here called RF list to matrix. So I'm converting the recombination fraction list to a matrix, right? So we are going to use this recombination fraction list to matrix. And if you do the pairwise estimation in this, using these functions, and you can also, and after that, you need to use this RF list to matrix. And yeah, so basically what I'm going to do here is to download this ready matrix for the whole genome. And it's available in this link here, but it's also available in the polyploids.org website. And I'm gonna load this. And here I have the recombination fraction matrix for the whole genome. And I'm going to print this. And as you can see here, oops, this is not the right thing to do. Oh, this one would take a while to do. This is not good. Yeah, so actually, I think this is better. Okay, so this is an unordered linkage a recombination fraction matrix, okay? And since we have the genomic order in our data set, what I can do is to use the function get genomic order in my sequence, and I will get the genomic order in that sequence, and I will plot the genomic order here, print the genomic order. As you can see, we have the name of the markers, linkage group or chromosome, and position in that chromosome. You can even plot this, as you can see, the distribution of the markers in your data set. And yeah, okay, there you go. Okay, so this is actually the linkage fraction, the recombination fraction matrix using the genomic order. And this is a very nice picture because we have a very good genome reference for potato. And as you can see here, it's pretty much ordered. And but still, even if we don't have this order, we can use the following functions to assemble linkage groups, and I'm going to show you how to do that. Okay, so this is a good linkage fraction matrix. You can see the 12 linkage groups of potato here, but now, even if you don't have that, which is the first matrix that I show you, I don't know if you saw the matrix, it's basically a blue matrix because the red dots are all around. What you can do is to use this group MAPpoly function. And in some cases, you have the expected number of the number of linkage groups. In this case, we have 12. And as you can see here, we have a very high concordance between our recombination fraction matrix and the clustering algorithm. And as you can see here, we have very distinct groups. And you can change to other number of expected groups. But I'm going to stick with this one. And the result of this group is this. So here I have all my 12 groups. So this is from what I just assembled using recombination fractions. And here, what I have is the genome reference. So here, my first group refers to genome, is connected to chromosome number 10. My second one with chromosome number 9. And why I know this? Because the majority of the SNPs are in this chromosome. And as you can see, there's some chromosomes, there's some markers of the diagonal here, and we can use those as well. And for some of the markers that were not anchored to any of the positions of the genome, we have this column here, okay? And again, everything that I'm showing you here is in this tutorial, okay? Good. So as I said to you, we can use MAPpoly function and we can use the group object here, and we can say, oh, well, let's just pick the first group. And that's what I'm going to do here. And this genomic information here that I have here is just to tell the program just to use the combination of the linkage and the genome at the same time, okay? You can just skip this and then it will just the linkage information. And here we have our sequence with chromosome number 10, and I can plot the recombination fraction matrix for this linkage group. As you can see, it's not ordered, right? But we can use two approaches here to order the markers. The first one is use the genomic order, right? So we are going to get the genomic order of sequence number 10, and we are going to plot this based on the genomic order. And as you can see here, the genomic order is a very good order because we have this monotonic matrix, and but also we can use the MDS algorithm here to do this job. So as Peter said, I also like to shut down the devices here because it's easier to do. And here, instead of using MDS map, we just use the functions that MDS uses, and we implemented the same things here with the same codes. And then instead of using the genome order, we can use the MDS algorithm. And there's some checkings that you can do. In this case, this is the NFIT values. So there are two very high SNPs here. We usually can remove those SNPs using this N argument here. And then we're going to plot this again. And as you can see, this is just a diagnostic plot. And if you want more details on this, just look into the tutorial. And if you have the MDS order, you can use your MAPpoly function to get the ordered markers for the MDS output. And then I can use the function plot in my matrix, and I can order these SNPs using the marker names in my sequence. And this is actually the output for the MDS algorithm, which is very, very good. Here you can compare the genomic order with the MDS algorithm. And as you can see, it's a very good fit. It's almost one-to-one here. It's impressive. And basically what MDS can't solve is the local ordering, right? But the big structure here, the whole structure of the genome, MDS captures. And this is remarkable. And okay, so we load our data set, we filter our data set, we computed the 2-point recombination fractions, split into linkage groups, ordered in one linkage group, and then I'm going to phase the markers for this specific linkage group. And the way we do this in MAPpoly is using this function strfHMM sequential. And there's a lot of arguments here. I'm going to run the function and explain the arguments because it will take like 3 minutes here. Yeah. So here we have our sequence of markers ordered. Here we have a start data set. In the start data set is the number of SNPs we start our analysis. it will perform an exhaustive search in this start set. Usually I put this 3 or 5 for both tetraploids and hexaploids. This is the threshold for how much the program should trust in the 2-point information. Usually I use 10 here, but you can try other values here. Usually, if you have smaller values here, they are more likely to happen errors in the phasing. And if you use high values here, you expect less errors in the phase, but it takes much more time. Because the indecisions that the program had here, it will be solved by the hidden Markov model process. And here I have another threshold for the hidden Markov model. So I have a sequence of a small number of markers, and then given the order that I'm providing to the function, I will sequentially add markers at the end of this chain. And if the 2-point function is not enough to solve, I'm going to rely on the HMM. And if the HMM is not enough to solve, here we have a log score of 50, it will be carried out to the next insertion of markers. And sometimes when you insert a new marker, you actually solve your problem. And then here we have something, an extended tail, which tells how many markers we should consider in the HMM analysis. Here you have your 2-point data set. In this case, I'm using the 2-point data set from the chromosome number 10. This is just tolerances for the convergence of the EM algorithm. The number of the phases that I'm allowing each one of the markers have. So for instance, if I analyze a marker, it has more than 20 possible phases, I will just drop this marker. And here we have something we call submap size diff limit, which is the limit that I'm allowing the map expands as I am insert a new marker. So if the map expands more than 5 centimorgans, I'm not going to use that marker, okay? So you can change this. Ideally, you should put as much phases here, as many phases here as you want, but unfortunately, this would make the algorithm very, very slow. And usually 20 is a good balance for the algorithm. And the last argument is info tail, which means that it tells to the algorithm to use the markers until he can distinguish between all homologs in that linkage group. So in this case, even if I choose to use just 30 markers to run the HMM, if I use info tail equals true, it will analyze if I can distinguish between all possible 8 homolog chromosomes here. And then just let me show you the output. So this is while constructing the map, right? So here, in this case, we have our sequence, our initial sequence was 3. In this case, we couldn't solve just with the 2-point analysis, so it evaluated 6 maps using the hidden Markov model. And at some point here, when we include this particular marker here, 137, it expanded the map, so we didn't include this marker. Here, in this case, we have too many linkage phases to analyze, so it just dropped this marker. And this is the final map. And at the end of this procedure, it estimates the multi-point linkage map using a hidden Markov model. You can print this map. Here we have the resulting map with every homolog in that linkage group. And here we have the distance in centimorgans. Here we are using Haldane mapping function. But you also can plot these markers in this graphic here. So here we have parent 1 and parent 2, the homologs. Here we have a color code for the dosage of the SNPs in a particular position. Here we have our traditional linkage map with the distance in centimorgans. You can also just plot a segment of this map using this function here. In this case, I'm using a limit of 38 centimorgans to the left and 42 centimorgans to the right. And here I'm also showing the name of the markers. And in this case, we can also use, so in the previous case, I used the genomic order, okay? And so in this case, in this first case here, I used the genome order, but we can also use the MDS order to do the same procedure. And in this script, I'm not going to use this here to show you here, but we can, because we don't have time, but you can run yourself this script here. And as you can see, you have a very similar map, but in this case, since we have these local inversions, the map will be inflated a little bit. And, but the results will be pretty much the same and the phase will be exactly the same, meaning that the algorithm is robust to small inversions. And in this here, so we have our order, in this case is based on the genome. But as I showed you in my first presentation, we have, sometimes we have errors and we need to remove those errors or at least to give some room for improvement, for the hidden Markov model improvement in this, in our final map. In this case, we can use two functions here. We have the function est_full HMM with global error, and we also have another function that uses the probabilistic input from MAPpoly or from other softwares. In this case, I'm going to use a global error, meaning that I'm assuming that I have a 5% global error of genotype calling, and I'm going to let the function to adjust the map using the hidden Markov model. And as you can see here, this will take a while. So each one of these interactions here is one of the EM steps in our hidden Markov model. And when you compare the resulting map here with the map we had with no error modeling, it's a smaller map because we remove some false recombination events which were caused by the noise in the data set, right? And this is done using the hidden Markov model or using the information in the whole genome that propagates from one marker to the other marker. So just let us compare here. I'm going to omit the MDS part here. I'm going to use this function, plot map list. So if you have several maps, you can put them in a list and just use this function, plot map list. And there you go. We have our map. This is a just simple representation of the map. So this is just with no error modeling, and this is using the error modeling. And if we have that map, the phased map, what we can do is to use this map to compute the conditional probabilities that are required for the QTL mapping. In this case, we use this function calc genoprob, which will run a hidden Markov model and will provide the probability of each one of the 36 genotypes for each one of the individuals in the whole genome. In this case, just for chromosome number 10, but in a step in 1 and 1 centimorgan. And when you do that, that's pretty fast. And when you have that information, what you can do is to use that for the QTL analysis, and then what we also can do here is to use this information to compute the probability of each one of the homologs in the offspring, and then we can build the haplotypes, the probabilistic haplotypes here. So I'm just going to run this for this chromosome. And here we have the results. So this is the result that I show you in my presentation, in my previous presentation. This is in Plotly. As you can see here, we have the probabilities for each one of the homologs in this specific individual for chromosome number 10. And we also can use, but this is just without the error, right? And then when you allow the Hidden-Markov model to correct these errors here, I'm assuming just a 5% of error, and I can do the same. And as you can see, it will remove this recombination, this false recombination points in my graphic. And here also what you can do is to draw genotypic preferential profiles. And this is for chromosome number 10 again. Using the conditional probabilities, I can compute the preferential pairing profile for the whole chromosome. And here, for this particular case, we have these possible configurations. As I showed you, in the case of the hexaploid, we had 15. In this case, we just have 3. And what we expect in an auto hexaploid is that they have the same probability. So let's take a look into this. And here you have, so the dashed line is the what you expect under a completely auto polyploid scenario in which every configuration here has the same probability. So we have parent 1, parent 2. We have a formal statistic test here to test if they are equal. And as you can see here, all configurations are have pretty much the same probability. Yeah, so here I have some, how can you use this using parallel functions. So we have, most of us have more than one CPU in our computer, so we can use multiple CPUs. I'm just going to skip this, but the code is here and you can try. And what I'm going to do here is to load the whole map. So this was done using this code that I'm just skipping, right? And in this case, instead of just showing you the results for chromosome number 10, I'm going to show you the results for all chromosomes. And in this case, what I did here, just really briefly, I did the same procedure that I did for the chromosome number 10 for all chromosomes, but I use a function that parallelizes this so we can use multiple processors in our computer. And at the end, what I did was to compute the conditional probabilities for the genotypes for every individual in every position for every linkage group for all 12 linkage groups. And what you can do here again is I'm going to show this. So this is just a data frame containing these probabilities for each one of the homologs and positions. But the most important part here is this. We can actually choose the linkage phase, the individual, the linkage group, and we also can plot this for all linkage groups for a specific individual. And as you can see here, this is the graphic for all individuals. So this is the probabilistic profile of the haplotypes for all linkage groups for individual, for this in 1, 2, 3. And what you can do again, you can compute the preferential pairing profiles for the whole genome. As you can see here, it's very fast once you have the genotype probabilities. Again, we have 3 possible configurations for each one of the parents. And here we have the probabilities. It's very close to 0.33, which is the expected. And then we can plot for the whole genome, right? And there we have it. There you go. So this is for all linkage groups. And as you can see, it's pretty much auto polyploids as we expect in potato. And here are just summary functions to export the map to publications or for your own record. So after that we did all this stuff, what we can do is to use this function update map that we reintroduce those markers that were redundant at the beginning. And then it will do this marker, all linkage groups. And this is the summary for the final map. Just let me draw this again. Oops, not this one. Yeah, so summary again. Yeah, so here we have just a summary of the map. We have the linkage group, the sequences present in each one of the linkage group. Here we have the map length. Here we have how many markers per centimorgan, number of simplex markers, double simplex, and multiplex markers for each one of the linkage groups, total number of markers, and total number of markers in the whole map, which is 4,740 markers. And this is the maximum gap in each one. So here we have something weird in chromosome number 10, in chromosome number 1. Maybe we want to check this, but we don't have time here. But this is a good way to inspect these gaps. And here is just comparing the genome order and the MDS order. But in this case, of course, since we don't have the MDF computed here, we are just comparing the genome order with the genome order, but we are comparing the genome position with which is in megabase, and with the map position, which is in centimorgan. So you have for all your 12 linkage groups. And if you don't want to, if you want to see all the groups compared, instead of just a one-by-one thing, because sometimes you have some markers that were, for instance, here we have this marker that belonged to chromosome number 7 in the genome, but it was mapped in chromosome number 4 in our map. So you can check that using this. You can plot a map list here, just with a regular map. And then you can export the final map to a CSV file using this. And use this export map list. And just to show you what we have here, I'm going to import this as we've had the. So here we have the name of the markers, we have the linkage group, we have the reference chromosome, we have the reference position within that chromosome, we have the reference allele. If you have a VCF file, it will show the reference and then alternate allele in these columns here. We have the map position, the dosage in parent 1, parent 2, and here we have the phase in both parents, right? So so this is the dosage in parent 1, parent 2, and these are the phases in both parents. So 1 means that the allele was present here, and 0 means that the allele was not present here. And with that, I'm going to stop here.
This presentation has been sponsored by the Tools for Genomics Assisted Breeding and Polyploids Research Project. For more information, you can email us at toolsforpolyploids@tamu.edu or visit our website at www.polyploids.org. This project is funded by NIFA within the USDA Specialty Crop Research Initiative funding program.